Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome to the next lecture in the first module of Plant Biotechnology course. In this module we are talking about the basics of plant tissue culture, which would include a variety of topics like totipotency, differentiation, somatic embryogenesis, organogenesis, micropropagation, meristem or shoot tip culture, and so on. My name is Manoj Sharma and I am working as an assistant professor of plant biology at Jawaharlal Nehru University, where I teach plant biotechnology and genetic engineering at School of Biotechnology. This lecture content has been reviewed by Dr. Kashmir Singh, who is working as an associate professor of plant biology at Department of Biotechnology, Punjab University, Chandigarh. This project has been sponsored by DTH Swayam Prabha, MHRD, New Delhi. In the first lecture, we had discussed about the cellular totipotency and basics of organogenesis as well as somatic embryogenesis. In this lecture, I will first briefly discuss about the major landmark developments in the history of plant tissue culture, and then I will discuss about the biological components or the parameters that influence the tissue culture responses. So, plant tissue culture is a collective term that refers to various procedures that are used to grow and maintain plant cells or tissues or organs on an artificial media under aseptic conditions and controlled environment. So basically it's a tool to exploit the cellular totipotency. In last lecture we learned about the totipotency, how each and every plant cell is totipotent, that is, it's capable to regenerate into a whole plant body. So it's a tool to exploit the cellular totipotency of plant cells for several applications in plant biotechnology, like micropropagation, that is the clonal multiplication of the plants, production of virus-free plants, crop improvement like production of haploids or polyploids like triploid bananas or triploid watermelon or grapes, which are the seedless. So these are triploid plants are seedless and we like eating these seedless fruits. Then it's also frequently used to conserve the germplasm or important germplasm. Somatic hybridization that helps us to bring the traits from two different species which are sexually incompatible by the fusion of these somatic cells. And then the genetic engineering based plant improvement. So basically the regeneration of whole plants from few cells or tissues is kind of a basic requirement for the genetic engineering based intervention of the crop improvement. Now this whole process of tissue culture, it includes several aspects or several steps, like how to grow experimental plants, how to select X plant. When we are talking about cells, tissues or organs, how to select which is the right tissue or which is the right organ, or how to select what is the right size of this X plant to initiate the cultures, how to sterilize or how to clean these X plants to initiate these cultures, or how to put this onto the culture media in a right orientation, choosing the right environmental variables like temperature, light, or various media components like the growth regulators or the nutrient components. So it's very important to carefully select these uh, uh, these uh, these steps, or okay, make the careful selections for the successful cultures. Hence, certainly it requires an experienced and a vigilant eye. Overall, we can say that the success of a large number of biotech-based applications in plant biology depends upon the successful establishment of the plant tissue culture for that particular species. Therefore, it's an exciting area actually in the plant biotechnology, in the establishing a plant tissue culture, and with several applications in basic as well as applied sciences. Now, let's have a briefly look at the landmark developments in the history of plant tissue culture. In the middle of the 19th century, we saw the development or concept of the cell theory, which was given by Schleden and Swan independently in 1839, 38 or 39, where they worked independently, looked at to cell, the microscopic structures, and they postulated that cell is a unit of structure and function in an organism. Several people started working after looking at cells in the microscope, and in the plants also people started working with the plant development. However, one major development was reported in 1878 by Watching, where he talked about the polar development in plants. Basically, he showed that the upper part of the stem segment always produce the buds or the shoots, whereas the lower part of the segment may produce roots or a callus-like structure. And it is irrespective to the size of the size of the segment from the stem that were taken to the culture. It was an important development where cell determination was talked about, that there is a bipolar development. Later, the theoretical basis of the plant tissue culture were proposed by the Gottlieb Haberland in 1902 for the first time when he was addressing to the German Academy of Sciences. So in the last part of 19th century, in 1890s, Gottlieb attempted to culture fully differentiated cells that were isolated from leaves, petioles, or any other fully differentiated tissues of the plants. But he was unsuccessful. He failed to actually culture them. However, based upon the experience to work on the topic for more than a decade, he learned several aspects of the plant tissue culture that were important for establishing successful cultures. So, he laid down several postulates or the principles about the plant tissue culture when he was talking in 1902, and he published these principles. Now, later, several other scientists examined his ideas about the plant tissue culture, about the postulates, what he had talked about, and confirmed them by experimentation. And later, he was given the title of the father of tissue culture. Soon after, within a few years, Herrings and Brown, he actually successfully cultured the embryos of the crucifers on the artificial media, that is the artificial solutions of mineral salts and sugar, and actually successfully cultured them to maturity. I mean, full-fledged plants were generated, and this was also the confirmation of what Gottlieb has said earlier in 1902. So then there were a lot of developments during this time. Several students of the Gottlieb Haberland did a lot of work in establishing plant tissue culture. They were able to establish the dividing root tip cells in the artificial cultures in 1920s or late 1920s. However, one major development was in 1929 when the Liebeck he rescued the embryo from an interspecific hybrid. So the interspecific hybrid was generated from two plants or two species which were sexually incompatible. Some embryos were formed, and these scientists, they removed the embryo from the plant, and they regenerated them on the artificial media, and mature plants were produced. So if these wouldn't have been rescued, these embryos would never have seen the maturity. Later in 1930s, Philip White, he worked with the isolated root cultures, and in 1934 White reported establishing continuously growing cultures of tomato root tips. They were maintained by subculturing onto the fresh medium for the longer period of time. In fact, he actually formulated the first synthetic media for the plant tissue culture, which was published in 1937 and was named as White's medium. It contained inorganic salts, sugars, and vitamins. During next 3 decades, several exciting developments were seen, like use of the coconut water improved the callousing efficiency significantly, or the callus culture for n number of species were established, even for the woody species or the herbaceous species which are actually recalcitrant, or for which the callus, tissue culture, establishing a tissue culture was a difficult job. It was successfully achieved. However, in 1950s, the discovery of kinetin or a cytokinin, it was a great milestone. Soon after the discovery of kinetin or a cytokinin hormone, the world had witnessed another landmark development in the field of plant tissue culture, and it was the chemical control of the root and shoot differentiation. That is, it was identified that is exogenous supply of auxins and cytokinins was able to greatly influence the morphogenic fate of the callus cells. It was and specifically these studies were performed in tobacco. So this was a huge development that now people knew that we can induce the morphogenetic responses in the callus, or we can induce the regeneration in the callus by kind of playing with the ratio of or the balance of these auxins and cytokinins. Another major development was the formation of somatic embryos from the carrot cells. It was also achieved by on the artificial media during in late 1950s. So this happened in 1950s, the late 50s. Now, by now it was well recognized that the media salts and growth hormones greatly influence the tissue culture, whether it is the callus initiation or the morphogenesis, induction or evoking the morphogenetic responses. And a lot of emphasis was put on to optimizing the media components like the media salts or growth regulators for culturing. Different kind of salt combinations were tried, different concentrations of these salts were tried, or different combinations of growth regulators or their concentrations were also optimized, and there was a lot of stress was put, or a lot of efforts were put towards optimizing these media. And in 1962 Mureshis and Skook, they published another media composition which is called as Mureshis or Skook medium, or it is also known as a MS basal medium. So basically they published the recipe. It was the detailed composition for all the media components of the salts that that were required to artificially initiate the cultures from or callus cultures from the explants. So it's called as the basal media, and even today it is this basal media, MS basal media, is used as a starting point for initiating the plant tissue culture studies or optimizing the plant tissue culture protocols for the species where these protocols are not available. In 1964, there was another landmark development and the contribution from the Indian scientific community. It was the successful establishment of the anther culture to produce the haploid plants. Contribution was made by the scientists from Delhi University, that is Shipra Gua Mukherjee and Professor S.C. Maheshwari, who could establish the anther cultures to develop the haploid plants in Dhatura. Then in 1960s, several development takes place, and people were now able to induce the morphogenetic responses and regenerated the full plants from the in vitro, starting from the explants, and then these protocols were replicated to several other species. Even in later part of 1960s, there is a huge development of the protoplast culture, protoplast preparation, and the protoplast fusion. What we discussed in the somatic, we will discuss later as in the form of somatic hybridization. These developments were made in 1960s, late 1960s. From 1917 onward, plant tissue culture evolved in the form of several different type of applications in almost every aspect of the plant biology research. Culturing of isolated cells helped to make progress in studying cell behavior, like the cytological studies, nutrition, cell metabolism, or the changes which appear in the cells during the morphogenetic differentiation. Meristem culture were optimized, which helped to develop the pathogen-free plant, and it proved to be very useful for germplasm storage. Clonal propagation was established at the commercial scale, and it is also one of the most widely used application even today. By mid-70s, it was worked out that agrobacterium transformed plants in order to cause the infection. That means that it transfers its small segment of the DNA into the plant. And this fact also kind of developed a hypothesis that agrobacterium could be used to transfer the DNA of our own interest into the plant genome. It means that the foreign DNA could be delivered to the plant genome, and in fact by early 1980s itself, transgenic plants were developed. This technology developed or opened up a totally new era of plant tissue culture applications for crop improvement, because for the genetic engineering studies, for any genetic engineering based or plant transformation based crop improvement, the availability of regeneration protocol was kind of a prerequisite. In India, plant tissue culture was established in 1950s at University of Delhi, and soon after several other centers started working with the plant tissue cultures. These included Bose Institutes, Kolkata, MS University, Vadodara, National Botanical Research Institute, Lucknow, were established. Similarly, several other universities later started working in this field, and later in 1980s, Department of Biotechnology was established, that also has one of the main focused research on the plant tissue culture and its applications in crop improvement or agricultural biotechnology. Next is the various factors that are crucial for the plant tissue culture. These factors are grouped in three broad categories, which are biological parameters, chemical and physical parameters. Biological parameters are the ones which are associated with the biological material that we use to initiate the culture. Or another way we can say that they are associated with the explant. Then is the chemical factors like the media salts or the growth regulators. And finally is the physical parameters that include light and temperature, etc. In this lecture, we will focus mainly on the biological parameters, and I will discuss the chemical and physical component in the next lecture. So let's start with the biological factors first. Explant, as we know, is a segment of the plant which is separated from the parent plant and is used to initiate the in vitro culture in artificial conditions. So the explant is the starting biological material, and it can be any living part of the plant. We can harvest any of the tissue. It should be living. That is the basic requirement. However, the physiological status of these living tissues vary from one tissue to another tissue or the organ to organ on the same plant. For better results, it is very important to choose the right explant. Though every plant cell is a totipotent, that is, it has a capability to regenerate a complete plant. However, the ease of this process, ease of initiating the in vitro culture from these explants may vary with the physiological status of these explants. And therefore, these mitotically or metabolically active tissues are the best source for the explant preparation. The responses, the in vitro culture responses from the metabolically or mitotically active tissues would be the best from among all explants. There are several different types of vegetative tissues that are in the different stage of metabolic activity and can be used to initiate the in vitro cultures, like meristems, apical meristem. It may be the root apical meristem or shoot apical meristem, axillary buds, cotyledons, the first embryonic leaves, actual leaves, roots or the root apical meristems, hypocotyles, inflorescence parts, anther, immature embryo. So all these tissues can be used as they are actively growing. However, the potentiality of these tissue would depend upon that particular species. So basic requirement is that one thing, basic fundamental thing that we need to keep in mind, that we need to harvest the explants from actively dividing tissues, which are, and it will be most responsive and best results will be produced. Now, potentiality of the same organ to use as an explant also varies with the species. It does not mean that if the one explant like the apical meristem or the cotyledon, if it is we are getting very good results in species A, then certainly it would produce good results in species B also. The physiological status of the same organ or the same tissue may be very different between the two species. So like for example, if we take the leaf tissue or the cotyledon tissue and we compare the monocots versus dicot. Now in tobacco or tomato, we can use the leaf segments to initiate the culture, and it's very successful. We use it, and they are frequently actually used in the tissue culture protocols. However, if we talk about the monocots like the rice or the sorghum, leaf segments do not respond at all it is almost it is very difficult to initiate or induce the callousing response from the leaf tissues of these plants. So the conclusion is that we should harvest the X plant from actively dividing and metabolically active tissues. Next is the health of the donor plant, the health of the plant itself, from where we are harvesting the X plant. So the physiological health of the donor plant has a huge impact on the regeneration potential of the X plants. Now what happens in the physiology of the plant development, it changes with the seasons. That is the environmental conditions around. Like for example, if there is a flowering plant, now spring, it flowers in the spring season. So this season would be best for its growth, and it will be metabolically very active during this time. However, the same plant in the winter season may not be metabolically active, or maybe metabolically dormant. And even if it is active, it will be growing at a very slow pace during these extreme cold conditions. We call these conditions as a physiological dormancy. So X plant harvested during the physiological dormancy would have low regeneration potential. So during the favorable growth parameters, plant grow actively, and X plants from these mitotic tissues would perform much better. However, during the unfavorable conditions, when the plant tissues are almost dormant, very little metabolic activity is there, same X plant would not produce the same result as it produced when it was harvested from the metabolically active state. So the physiological state of the same organ also varies with the season. So it is very important to look for the right season and right environmental condition for harvesting the X plants. Now, stress on the plants also compromises the regeneration potential. Yes, that's fine. We are talking about that we should look for the right season, like the spring season is best for this flowering plant that we were talking about in the last example. However, during the spring season also, there may be some other stress factor which is limiting its growth, like maybe there is not enough water for this plant and there is a drought-like condition. So now these conditions, these stress conditions, compromise the metabolic activity of or the growth and development of this plant, and this plant is stressed. And so these stress plants also, the X plant harvested from these stressed plants even during the season when these plants grow best may not produce the good results because of the compromised status of the physiological activity of these plants. And so we need to make sure that plants are actively growing, they are healthy, and they are not stressed. So in this way, if we talk about the greenhouse grown plants or the plants grown in the culture room where conditions or external conditions are controlled, they perform better. So the X plant harvested from the greenhouse grown plants, they perform much better as compared to the field grown plants. And even like aseptically grown seedlings, X plant will perform even better than the greenhouse grown plants. So aseptically, in vitro seedlings, they produce the best results. Now the genotype of the plant. Now it's also well-known fact now that the regeneration or the somatic embryogenesis or the organogenesis is a genetically controlled trait. It is genetically regulated. Striking differences in the regeneration potential among the various varietal lines or the intravarietal lines or the intervarietal lines have been reported in the past. So the different cultivars of the same species may respond differently, and certainly different species may respond very differently though they belong to the same genus. Now these differences may be because of the differences in the endogenous levels of these growth regulators. Now growth regulators, they are the integral part of the plant development or morphogenetic responses. They are naturally produced in the plants, which result in the natural differentiation, that is a root and shoot differentiation and the differentiation of all other tissues. And so these hormones, they are there, they are produced. Their concentration may vary from one tissue to another tissue, and hence the endogenous level or endogenous concentrations of these hormones or the growth regulators, it can also impact the response of these explants to the tissue culture. It has been found that the cultivars or the varieties that usually have higher embryogenic potential, higher somatic embryogenic potential, they usually have lower level of phytohormones in their ovules. So basically endogenous level of the phytohormones in the explant would also impact the response of these explants to the in vitro conditions. Now this Brassica is a very good example to describe the genetic control of the regeneration process. So Brassica oleracea is we know it as a cabbage or a cauliflower. It has a very good regeneration potential. It means that the regeneration protocols are easily available, and it is comparatively easier to establish the cultures or in vitro cultures from the explant harvested from Brassica oleracea. However, Brassica campestris, we know it as a Chinese cabbage or turnip, is least regenerative among the Brassica species. So what does that mean? That means that if it is difficult to initiate the in vitro cultures from the explant that were harvested from Brassica campestris. Now there is another Brassica species, Brassica nepus, which is an alloploid generated from these two cultivars, two species, Brassica oleracea and Brassica campestris. And if we see the regeneration potential of this Brassica nepus is intermediate between the regeneration potential from Brassica oleracea and Brassica campestris. So this performed better in the in vitro cultures as compared to the Brassica campestris. However, Brassica oleracea performed better than this one. So this clearly describe or explain the genetic regulation of the regeneration. So the genotype itself can influence the genetic or the response of the explants towards the in vitro cultures. So therefore, many times when we are establishing the cultures for the first time for any one particular species, and we should start with the multiple cultivars. It's many times it's possible that we pick a cultivar that is very recalcitrant to the regeneration response, and hence it may not respond, or it will take longer time to establish, to identify or to optimize the cultural conditions for these ones, rather than if we find a cultivar which is responsive or better, which produce the better results. So the genotype do have a very important or significant impact on the regeneration potential of the plants. Then is the size of explants. So generally the explants which are very small, they are harder to culture. Like if you want to establish the culture from a single cell or the protoplast, they are more difficult to establish as compared to when larger segment of the plants are used, like if we are using the leaf segment or cotyledons as the explants. So initiating the callus will be much easier as compared to when we are using the single cells or the protoplast. And it is probably because the larger plants may have the larger nutrient resources or the hormonal reserves, hence they perform better in response to the in vitro cultures. Another important component is the contamination, that is the source plant from where we are harvesting the explants excising the explant, they should not be contaminated with the pathogen. Now, infection is actually a stress on the plant. It compromises the physiological status of the explant. It compromises the metabolic activity, growth and development. And in addition, if your explants are contaminated, they were difficult to clean, and they may contaminate your cultures also. So it's very important that our plants, from where we are harvesting the explant, they should not be contaminated from the pathogens. In addition, the location of the explant on the source plant also matters, specifically if they are field grown plants, or if they are they are field grown plants. Like if the explant are harvested from the locations closer to the soil, they are harder to clean. There may be more infections on these tissues which are closer to the soil as compared to the tissues which are away from the soil on the towards the tip of the plant. So it is always good idea to harvest the explants which are from the tissues which are located at the position away from the soil in the plant. So typically aseptically grown seedling would be the best source, and this location would not impact in that condition. Next is the orientation on the media. Now, orientation of the explant can also influence the tissue culture. The orientation means how we are placing the explant on the media. Now, this is a tomato seedling or tomato plantlet. Here we see the abaxial surface, that is the lower surface of the plant is the abaxial surface, and the upper surface of the plant is adaxial surface. So if we are using the explant segments from the leaf tissue, we can either place the abaxial surface touching the media, that is the lower surface touching the media, or we can place these leaf segments upside down, that is the adaxial surface touching the media. However, it has been found that response of these explants are much better when they were placed with abaxial side touching in contact with the media, that is the lower surface of the leaf touching the media, they perform better. Even like twofold differences or a twofold callousing responses has been reported just because of the orientation of these explant in some species. So today in this lecture we learned about the landmark developments of plant tissue culture and various biological parameters of biological material that greatly influence the tissue culture process. The parameters that I discussed today were explant health, health of the donor plants, genotype of the source species, explant size, contamination or the orientation of the explants on the media. And in the next session, I will discuss about the chemical and physical parameters influencing plant tissue cultures. Thank you.